In this video, we'll talk about some basic mutations that can happen in DNA. So up here in black, I've written template stranded DNA, and I then transcribed that into mRNA. You can double check that for me to make sure I didn't make any mistakes. G-A-U-G-U. I think that looks good. Just forgot to put my 3' over here. I then translated that mRNA. Again, I'll double check to make sure that lines up. Yep, okay, I think that's right. And now we'll look at 3 scenarios of how that DNA could change. So when DNA is replicated, DNA polymerase once in a while will insert the wrong base. Right? It will insert the nucleotide with an incorrect base. And there are mechanisms to repair that and to proofread and check for that. But so once in a while they'll slip through undetected, and they can then be used as template DNA and create different proteins. So remember, if the template DNA sequence is changed, the mRNA sequence will change the ordering of the amino acids, which will change the three-dimensional shape of the protein, which will change the functionality of that protein, possibly. So a change in the DNA can be a pretty big deal. And if this happens in a germ cell, a cell that's like a sex cell that's passed down, then that mutation will be a part of the offspring. Okay, so let's look at these scenarios. So in scenario number 1, this CAC is turned to a CAG, okay, in the DNA, which means the mRNA will go from GUG to GUC, which means the amino acid will go from Val to Val. Right? So we ended up changing the DNA, but because the genetic code is redundant, we actually had no change in the actual amino acid. That's called a silent mutation. That kind of makes sense, right? There's a mutation, but it actually ended up not having an effect on the protein, right? From the protein point of view, there's no difference. It's got all the amino acids all in the right order. Let's look at situation number 2. In situation number 2, I go from CTA to CTT, which means my mRNA becomes GAA instead of GAU, which means my amino acid becomes GAA glue instead of ASP. And this is called a missense mutation. Missense mutation. And that's because the amino acid changed. Whereas in the silent mutation, there's no amino acid change. Amino acid change. And let's look at number 3. At number 3, we have a ACA that becomes an ACT, and then that will make the MRA into UGA, which will give us, if you look on the amino acid sheet, a stop codon. Stop. And this is called a nonsense mutation. So these are the 3 kind of main types of these kind of simple mutations. This is called a nonsense. It's nonsense, right? It's because it just stops before it's really supposed to finish. And that's when an amino acid is changed to stop. So there you go. You've got your silent mutation where there's no change in the amino acid. You got your missense, which changes the amino acid, and you got your nonsense, which changes the amino acid to a stop. And this nonsense mutation can be a really big deal. It can totally change the protein, whereas the silent is pretty benign and the missense is kind of